Just a couple of quick announcements before we start. Here's the schedule for the rest of the summer. So we actually have another talk next week. So Sebastian, up there in the middle, is going to be talking to us about microscope objectives next week. Okay. And then down here, there's nothing on July 20th. But now there is. So hopefully you saw the email that I sent out last night. We have a software company called VisioFarm. They're going to be coming and doing a full-day workshop. So we have room for, I think, about 23, 24 people in the computer lab in Northwest. We're going to start at 9 in the morning. They're going to take us through all the different features of their software. You're going to be able to follow along on your own workstation and click through the software. This is an evaluation that we're doing. I know a lot of people have been having difficulty, especially with axioscan files. So you have these really big, large files from your multi-slide scanning, and sort of wondering what to do with those, how to quantitate it, how to handle them. I think this is a pretty good solution for it. So I had a quick tour of it a few weeks ago. I think it might be a good option, but we're hoping that a lot of people can come in and try it out and give us some feedback on whether or not you think it's something that's useful for the facility. So if you're interested, there's an email that went out to our listserv last night. It's got some instructions on how to sign up for that. And if you've got any questions, just send me an email. All right? And then we'll be back here again on the 27th. Just take note that we're going to be starting a half hour earlier that day. We've got a small demo that we've got to run in the facility in the morning, so we needed an extra few minutes. Okay. So that brings us to today's topic of fluorescence lifetime imaging. So why are we doing this talk? We were fortunate enough to come across a grant well over a year ago now to buy some Flim equipment for the facility. I'm excited to say that after a very, very long wait, this is finally going to be installed on the 15th. So as of July 15th, we're going to have the capability to do fluorescence lifetime imaging in the facility. A lot of you might not know what Flim is, so I thought this would be a great opportunity to introduce you to it. Just a word of warning, I'm by no means a Flim expert. I've done a little bit of lifetime measurements in a totally different capacity than what I'm going to talk to you about today. But this is just going to be sort of a general overview of what you can do with the technique and how it works. Okay, so this is our outline for today. We're just going to start with a review about fluorescence, how fluorescent molecules work, and how we take advantage of that in fluorescence lifetime imaging. I'm going to talk a bit about the hardware setup for Flim, so the Flim equipment itself, as well as what has to happen on your microscope. And then I'll just wrap up by giving you some examples of some common Flim experiments. Okay. So first of all, I'll just point out that Flim, like everything in microscopy, goes by many names. So Flim stands for fluorescence lifetime imaging microscopy. Sometimes this is also called TCSPC, which is time correlated single photon counting. And in the materials world, this is often referred to as TRPL or TRPL, time resolved photoluminescence. Essentially, they're all the same thing. So how does fluorescence work? So this is a bit of a review of, let's say, basic physics, basic chemistry from undergrad and late high school. So here is our horrifically out-of-date diagram of an atom. So we've got our nucleus in the middle, and we've got some electron shells around the outside. We've got our one little electron here that's spinning around our nucleus, all nice and happy. All of a sudden, out of nowhere, a blue photon of light comes in and smashes into it. Okay? And what happens is that electron absorbs some of that energy and moves to a higher excitation level, a higher orbital. Right? And now, while he's out there, at some point, he's going to release that energy as another photon of lower energy than the original photon. So you see this was a blue one, this is a green one, so it's got lower energy. And then he's going to return back to his grounded state. Okay? So this is what happens in a fluorescent dye. Obviously, there are many, many things wrong with this diagram. One thing, we know that electrons don't just spin around nuclei. They live in these orbitals that are more probability centers of where your electrons are likely to be. And it's just kind of moving between these different energy levels. Okay? Of course, there aren't many single atoms that can do this. This is usually done in a molecule, so these diagrams become much more complex. But if you think about a single fluorescent dye, so let's just think about something that we use all the time, like GFP or Alexa 488. It has some unique properties to it. So obviously, one of the unique properties are its chemical structure. So this is an example of Alexa 488. What are some other unique properties of fluorescent dyes or proteins? Exactly. So they're absorption and emission properties. Actually, that's the next one. Okay, so each one has this characteristic absorption spectra, which you can see by the blue line here, and a characteristic emission spectra, which is the red line. Is there anything else that's unique about a fluorescent dye? Trying to make this week interactive instead of just talking. You and I are on the exact same page here. Pretty sure that's, yeah, exactly. So quantum yield and extinction coefficient. These are some numbers that we attach on top. Okay? So extinction coefficient is essentially the ability of a dye to absorb light. So what's the probability that when a photon comes into a dye that it's actually going to interact with that electron shell and be absorbed, right? These are usually just a whole bunch of big numbers. You can measure these yourself. But you can see each dye has its own characteristic extinction coefficient. The higher this number is, the better that dye is at absorbing light. Okay? And these are all measured at a specific wavelength. And with the Alexa dyes, it's usually right at the wavelength that's in the number of the dye. Okay? Each dye also has a quantum yield. And now the quantum yield is the likelihood that a fluorescent dye is going to release a photon if it absorbs a photon. Okay? So not every photon that gets absorbed gets released as another photon. There's other ways to get rid of that energy. So the quantum yield just tells you the likelihood that an absorbed photon is going to be emitted after absorption. So what you can see is that a dye might be really good at absorbing light, but not very good at re-emitting a photon, or vice versa. So these numbers alone don't really tell us a lot about, they aren't the best way to find the perfect dye. So does anybody have a really simple idea of a better measure to sort of combine both of these? You might see it sometimes in charts referred to as brightness. What's one of the easiest things you can do with these two numbers? Multiply them. Good job. See, we're getting this working. Okay, so brightness is the extinction coefficient multiplied by the quantum yield. Right? And this gives you a much better way to compare dyes to one another. And if you look down here, 3 of these dyes are ones that I commonly recommend to you in the facility. For one reason, because they match the laser lines on most of our microscopes very well. So if you're doing a 3-color experiment, I usually suggest you use 488, 568, and 647. Now, along with matching our laser lines really well, you'll also see that these 3 dyes have 3 of the highest brightnesses. Okay. Alexa Fluor 546 is also excellent if you're using one of our older confocals that has a 543 laser line, or the 700 that has a 555 line. Cool. So now there's one more unique property And that's what we're going to talk about today. And this is sometimes a little bit harder to find. It's often not in all of the charts out there, but there's something called the fluorescence lifetime. I'm going to tell you a bit about what that is on the next slide. But what you can see is usually measured in nanoseconds, and each one of these dyes has a unique fluorescence lifetime. So what is a fluorescence lifetime? Anybody have any idea? Yeah.
How long is before it gives off like the next photon?
Yeah. You were listening in MTV 68.
Yeah.
Great. So essentially, the fluorescence lifetime is a measure between when a dye is hit with excitation light to when it gives off a photon. Okay? So the way we often describe this process is with this really complicated diagram called the Jablonski diagram. And for the most part today, we're just going to concentrate on one little area here. So let's just look at this red box right here. Okay? So most of the time, our fluorophore, our dye, is residing in the ground state. So those electrons are in their lowest energy level. Okay? When a photon comes in that matches the absorption spectra of that dye, it can be brought up to an excited state. Okay? And these are called the excited singlet states. And there can be multiple of these. This diagram looks different for every dye. And so we have a lower energy one called the S1 and a higher energy called the S2. So this process of being hit with that excitation photon and going to the excited state is a very, very fast process. Okay? So you can see the green line here happens in a femtosecond. So that's 10 to the minus 15 seconds. Okay? So this is a very, very fast process. Now the next thing that happens is this dye is now in an excited state, and what it has to do is it has to get back to the lowest level, lowest energy level in the S1 singlet state before it can actually fluoresce. Okay? So this is usually energy that's given off by vibration and heat. Okay? So what it's going to do is it's going to slowly tumble back down until it gets to the bottom of this S1 energy level. Okay? So this process is still pretty quick, not as quick as the excitation. This takes about a picosecond. And now, once it's down at this lowest energy level, at some point, it's going to release a photon of lower energy light than the photon that originally excited it. Okay? So that's the red line here, coming back down into the ground state. And this is the longest process. So sort of sitting at this bottom energy level, and this is where you have a nanosecond or more delay. So the entire process only takes nanoseconds. This is pretty much your limiting step, waiting for this photon to be released. Okay? So your fluorescence lifetime for a dye is the time between when it's hit with this excitation photon until when this emitted photon is released. So this is just showing this in a diagram. Essentially, we get absorption, relaxation, and then fluorescence. Okay. So if we wanted to measure this, how are we going to do that? The first thing we're going to need is some way to excite that fluorophore, right? So in the facility, what are some things that we use for exciting our fluorophores? Lasers, yeah, that's one. What else do we use? LEDs, yeah. So primarily we use lasers or LED lamps, or metal halide lamps or mercury lamps. So what happens if we were looking at a single molecule, you can say Alexa-488, just a single molecule of Alexa-488, we turn on our LED lamp, shoot some blue light onto it. It's going to absorb that light, it's going to relax, it's going to give off some fluorescence, and then what's going to happen? Is that light still there? It's just going to repeat this process again, right? It's going to go back to the start and start again. Unless what happens? Bleaching, the one thing you always hate, you don't want to happen. Okay, so if this is just doing this continuous loop, how are we going to measure a lifetime? If we take one of our lasers, so most of the lasers that we have in the facility are what are called continuous wave lasers. So it's just a steady stream of photons coming out of them. Does that change this loop in any way? No, right? So how are we going to break this loop? How are we going to stop this loop? How are we going to have a definite start, a definite stop point that we can actually make a measurement from? Something that we can do with light from either of these, actually. So if we didn't have a continuous wave of light, what could we use? This is what's common in all your two-photon lasers. They're all special type of laser. Pulse, exactly. Okay? So in the case of an LED, we could just flash that LED on and off really fast. And in the case of a laser, we can use a pulse laser, again, just packaging little packets of photons in time. And so now what that introduces, it's on another slide coming up, but it's essentially going to introduce a little delay right here. Okay? So, to do fluorescence lifetime, what you require is pulse excitation light. And you also require those pulses to be in the picosecond to nanosecond range. Okay? So these are some pulsed LEDs that comes from a company called PicoQuant, which is who we bought our Flim equipment from. And these are the best ones that they sell. And you can see that the pulse widths are in the range of about 1 nanosecond. This can be used for Flim, but it's not ideal. Okay? So in general, we don't use pulsed LEDs for this. They just aren't quite fast enough right now. But if we take a look at a pulsed laser, here's what we have happening. So this is the power being measured coming out of the laser head over time. And what you can see is in a continuous wave laser, this would just be a flat line along here. But with this pulse laser, what we have are little pulses in time. And there's a couple measurements that are important here. And you'll often see them listed on the specifications for a laser, for a pulse laser. You'll see something that's known as the peak power. So that's what's the highest power level that it reaches at the top of each one of these pulses. You'll also see the average power, which is just the power integrated over time. And this is much, much less. And so this is why when you're doing two-photon imaging, you can use a really, really high power laser that has a really, really high peak power that would usually just melt your sample and destroy it. But because it's being packaged in time into small little bits, the actual average power delivered to your sample is quite low. So those are two important specifications of the pulse laser. And the other two really important ones are the pulse width, so how wide in time are each one of these pulses, and then the pulse frequency, so how often do these pulses come. So if we look at most of the lasers, pulse lasers that we have in the facility, most of them are 80 megahertz. So that means you're getting 80 million pulses per second. And that represents about 12.5 nanoseconds in between each pulse. And then our pulse widths are all between 0.1 to 100 picoseconds. Okay? So what this means is when this laser is running at 80 megahertz, this delay is about 100 times longer than the pulse width. And it's 3 to 10 times longer than the fluorescence lifetime of most of the dyes that we're looking at. Okay? There are some dyes that do have longer lifetimes than that. Also, if you start getting into things like looking at phosphorescence or some types of autofluorescence, things do get longer. Some of the dyes, or some of the lasers that we have, the pulse lasers, you can actually change this frequency. You can go down to 50 and I think even down to 20 megahertz. So what that does is it ends up expanding this delay even longer. Okay. So these are the systems in our facility that currently have pulse lasers. So the 880 number 2 system, this is where the Flim equipment is going to be installed first, starting next week. So it has a 405 laser and a 440 laser that are both pulsed. Right. In addition, the 2 photon system, we actually just found out today that the new one should be here in September or October. It will have the pulse 440 laser and as well the 2 photon laser, which can, in theory, excite most dyes that you're going to be using, is also pulsed. So although it looks like we can't excite that many dyes because we only have these near UV and blue lasers, this tunable laser can excite quite a number of dyes. So we do have the ability to measure Flim on a number of different materials. Okay, so that's the excitation. The next thing we have to do is we need to somehow detect. So we actually have to detect these pulses with a way that we can actually time them. So now we come back to our absorption, relaxation, fluorescence, and then we've instituted this 12.5 nanosecond delay. So we get a pulse of light that's absorbed, we get relaxation, we get fluorescence, then we have a 12.5 nanosecond delay, and then we do this all over again. So obviously 12.5 nanoseconds is a very short amount of time. And because of that, that really limits the detectors that we can use for this. So often in the facility, if people say, what's the microscope you can image the fastest on? We'll say the cell observer or the axiozoom with one of these Hamamatsu SCMOS cameras. Okay? This is a camera that can read out the entire field of view. In 100 frames per second, you can do a smaller field of view in 800 frames per second, I believe. So it's really, really fast. But that's not fast enough for doing this. Okay? So photomultiplier tubes, which we have on our confocals, we never usually associate that with being fast because it takes so long to take an image on a point scanning confocal. But over here, this is a parallelized detection. You're imaging all of the pixels all at once. With a photomultiplier tube, you're imaging each pixel one at a time. So although this takes a second or two to image your entire field of view, you've imaged 4 million pixels in that time. So you've actually taken 4 million different measurements. Whereas this might only take 20, 30 milliseconds, but you've really only made one measurement, right? Just at multiple different speeds. So these photomultiplier tubes are actually really, really quick. They can be run quite fast. And they're predominantly what's used in Flim detection. Okay? There are some cameras out there that you can do this with, but it actually is basically a modified form or a multiplex community. So just a quick review of how a photomultiplier tube works. So the other thing that I didn't mention that's really, really important for this, you basically have a single photon hitting your detector. And that's not going to generate enough electronic signal for you to actually register it back in the electronics. So you also need to be able to multiply that signal multiple times. So this is another reason that photomultiplier tube is much preferred to a camera as well, because you can get much more multiplication. So this is a diagram of what a photomultiplier tube looks like. What we have is we have our photon that comes flying in, and it hits the photocathode. So this is a material, in our case it's going to be gas, so gallium arsenide phosphide. But when a photon hits it, it releases an electron. Okay? That's a property of the semiconductor, of the material that we're using. So that electron then flies into a vacuum tube, and there's an electric charge that's placed across this tube. And what it does is it draws the electron that's been created here at the photocathode to the anode. And there's some focusing electrodes, and then there's these little metal plates that are called dynodes that sit all the way down here. And what happens is that electron bounces off each one of these dynodes as it goes down. And what happens here is whenever an electron slams into one of these pieces of metal, it releases what are called secondary electrons. So what you're getting is a multiplication at each one of these little metal plates. Okay? And so we're going to be using detectors on the two systems that I showed you earlier that have just been recently released by Zeiss. It's called the Big 2 detector. It's actually a Hamamatsu gas PMT inside it. And this is going to give you about a 10 to the 8 electron gain. So from one photon, you're going to get about 10 to the 8 electrons out of it in the end. And they also have a time resolution of about 200 to 300 picoseconds. Obviously, this is really important. You have to be much less than that. You have to have a time resolution shorter than the lifetime of your fluorophore. And one of the big contributors to this time resolution is actually the length of this tube, the amount of time it takes that electron to fly the length of the tube. It's one of the key limiting factors. Cool. So we've got our excitation taken care of, we've got a pulse laser, we've got our detection taken care of with a really fast PMT. And the last thing we need is some instrumentation to handle our computational electronics in between. So what we're going to have is one of these big electronics boxes from Picoquant, and this essentially handles all the timing. So this box gets a signal from the laser when the laser fires that pulse, and then it collects all of the signal coming off the detector and puts those into little time gates. Okay. And sort of syncs all of that all together. And then, of course, we need some software so that we can interact with this and actually display our data. So those are the last two important things that we need as far as equipment goes. So what does one of these measurements look like? So you saw a little bit of an example of it down here. Most Flim data that you're going to see is just going to look like this simple graph right here. You're going to get a quick uprise and then a slow exponential decay. So what is that? Those graphs, for the most part, have the intensity, or the y-axis here is called the intensity axis. And this is, you can think of it much easier, it's just the number of photons that are in the detector. And then along here is the time axis, which is usually in nanoseconds. And this represents time after that initial pulse from our laser. Okay? So what happens is our laser fires a pulse. We get excitation of our fluorescent dye. And that, then we have relaxation. And we eventually release a photon, which is then detected by our detector. Okay? And our detector says, I detected that photon 3 nanoseconds after that initial pulse. And we put a mark there. So this just becomes a histogram. We fire our second one. Oh, that one was 1 nanosecond. Okay? And you can see we just do this over and over again. And what you'll essentially get is this curve. Okay? So we just have a histogram with an exponential decay. Now, the truth of the matter is we are analyzing, I was going to say imaging, but it's not really imaging, the average lifetime of a fluorophore. Okay? So each one of these events that gets detected has a slightly different lifetime. Okay? And there's a number of reasons for that. Some of it is our ability to measure. Also, each electron spends a little bit different amount of time in that excited state. Okay? So we get one of these exponential decay curves, and then we basically look for the 50% point, and we give that as our lifetime. Okay? So don't worry about the second curve. We'll talk about that in a second. But essentially, you have your intensity here, time here. At some point, we give it a characteristic lifetime for our dot. Now, sometimes you see these as shown as the exponential decay. This is usually a mono-exponential. And so what that means is you can also plot this on a logarithmic y scale, and then you'll actually see a linear decay. So on some of the ones that I'm going to show you a little bit later, you won't see an exponential decay, you'll actually see a linear decay. So just to give you a heads up. Okay. So that's the hardware and how the system works. So what do we actually do with this? So there's a number of different applications that fluorescence lifetime imaging has been used for. If any of you have done multispectral imaging or linear round mixing on our confocals, so if you have some autofluorescence in your sample that you're trying to get rid of, or you're really packing in a lot of dyes and you've got some bleed through between those dyes and you're trying to separate those out, you can essentially do the same thing with fluorescent lifetimes. As long as each of those dyes or your autofluorescents have a different lifetime, you can bin that out. So I'll show you an example of this a little bit later. Okay. Another thing that's often used is just looking at autofluorescence in different tissues. So this is actually the example of a retina. The tissue in the fovea, where the optic nerve comes in here, has autofluorescence with a different lifetime than the rest of the retina. So this is actually sometimes used in the surgical suite as well for different applications. In material science, this is quite often used for characterizing new materials, particularly fluorescence ones, so you can actually get a fluorescence lifetime as a characteristic of your new material, especially if you're working with fluorophores or say quantum dots, something like that. Sometimes also used for quality control in semiconductors, solar cells, and actually in laser diodes as well. And what you can see is you can look for the homogeneity of fluorescence across your product that you've made. But most often in biology, it's used for one of these three techniques. So looking at molecular interactions, sensing the environment, so oxygen, pH, calcium, and conformation changes in specific proteins. And these three are usually done with a FRET assay. So I'll explain FRET in a second. But before I get into FRET, I will say this sometimes doesn't have to be done with FRET though. You can tag a GFP molecule onto your protein of interest, and sometimes, definitely not all the time, if that protein, let's say it's a kinase that goes through a conformational change when it's doing a phosphorylation step, that conformational change can actually affect the lifetime of the GFP molecule. It just depends on where it moves and what it comes into contact with during that process. So this doesn't have to be a FRET assay. The majority of the time it is, but there are some molecules out there, if you stop a fluorophore on them during their computational changes, whether it's binding to calcium or some other ion in your cell, or actually making a structural change, that will affect the fluorescence lifetime of the fluorophore that's plugged on there. Right? It doesn't happen often, but when it does, it's fantastic because it's a really easy assay. And a lot of you probably have a bunch of proteins of interest in your lab that are already tagged with fluorophores or fluorescent proteins. Okay, so let's just talk real quickly about FRET. I feel this is something that most people are familiar with, judging by the number of people that ask me about it. But what people aren't familiar with or haven't heard of are the pitfalls of FRET. And there are a lot of pitfalls of FRET. So just a quick review what FRET is. It's a transfer of energy from a donor fluorophore to an acceptor fluorophore. And what that means is that the emission spectrum of your donor overlaps with the absorption spectra of your acceptor. So usually what happens is, let's say this is your donor, some blue light comes in, it usually gives off a green fluorescence. Right? And if these molecules are pretty far apart, that's what's going to happen. If they get close enough together, and there's that overlap between the emission of this one and the absorption of this one, what's going to happen is that blue light is going to come in, and instead of giving off green fluorescence, it's going to transfer that energy over to the acceptor, and the acceptor molecule is going to go into its excited state, and then it's actually going to give off, let's say, red fluorescence. Okay? So in this situation, instead of blue light coming in and green light going out, you have blue light going in and a transfer of energy, and then red light going out. So this will only happen if these molecules are in very close proximity, so 1 to 10 nanometers. Just know that a protein itself is about 10 nanometers, 9 to 10 nanometers, 8 to 10 nanometers in size. So these things really have to be right up against one another. Okay? And there's 3 primary methods that are used for detecting FRET. So the first one is essentially what I just described to you is sensitized emission. So this is taking your system that has both donors and acceptors in it, shining in some blue light, and looking for red light. There's a lot of problems with this. There is a ton of crosstalk in these systems, and the amount of red light that's coming out is so minuscule. This is a very, very difficult assay to do. To be honest, I don't always believe it when I see it. That said, I have published a paper with it, so I guess I can't be too accusatory. The other one is acceptor photobleaching. So the idea here is you take an image of your sample with both your donor and acceptor in it, and then what you do is you bleach your acceptor so it can no longer receive this energy from the donor, and then you take that image a second time. And what's actually going to happen is this donor will now be brighter because it's no longer transferring its energy to the acceptor. All of this energy is coming back out in this green light. Okay? So this is a method that's commonly used, and in general it works quite well. Okay? So usually if I ever see people, I say, do at least 2 of these. But now what we have is this option of Flim FRET. And in my personal opinion, this is the best way to do FRET because it takes out a lot of the general issues or problems with FRET. I'll get to that in a second. So what are we doing with Flim FRET? Here's an image of an experiment. So this is just a cell expressing GFP alone. And then this cell here is expressing a fusion of GFP and M-cherry. Okay? So it's just 2 fluorescent proteins fused together with a polypeptide linker. And what you can see is the GFP alone has a lifetime around 2.6 nanoseconds, kind of in the yellowy-orange range here. But when you fuse it to M-cherry, so this is only looking at the GFP lifetime. This is not looking at M-cherry at all. You can see this now drops down to about 2.2. Okay? So if we look at our correlation curves over here, this one's plotted with a logarithmic axis, so now we're seeing linear curves instead of that exponential decay. And you can see what's happened is this is just shifted to the left. Okay? So here's the lifetime in green. These are the lifetime measurements in this situation when you just had GFP. And this red line is the lifetime measurements when you had GFP fused to M-cherry. This is what you see. It's just this characteristic shift. If you're looking at exponential decays, it's a shift to the left. When you're looking at linear, it's more of a shift sort of down to the left. This little block line here, something I haven't talked about yet. This is called the IRF, the Instrument Response Function. And this is a measurement that you do at the beginning of every Flim experiment. And this is actually just looking at your reflected laser light. Okay? So this is showing you the actual pulse width of your laser. So this one here is pretty light. So if you ever see IRF, that's what that is. Okay. So why does this shift to the left? This is a question that kept me up all night last night, and actually had to do the old millionaire phone a friend to figure this out. Okay, because essentially what we're doing is we're only looking at our GFP. So why is the lifetime getting shorter? I understand that a lot of the energy is going over to our other molecule, the M-cherry, but we have no way of measuring that, right? We're just looking at GFP fluorescence. So why is this getting shorter? It's actually pretty interesting. Anyone know? Anyone want to take a guess? This one's tricky. Okay, so here's our Jablonski diagram for GFP. Usually we have a blue photon that comes in, pumps GFP up to its excited state, we get some vibrational relaxation, and then eventually a green photon is given off, and we fall back to the ground state. But when we have a molecule of M cherry in close enough proximity to GFP, there's something else that can happen. All right? So here's our M cherry. It's a red-shifted protein, so it's a slightly lower energy level than GFP. And now we have two pathways. There's actually more than two, but we're just going to talk about two. Two pathways out of the excited state. So we can give off a green photon and fall back to a lower energy level, or we can transfer that energy over to M cherry. Okay? So this is transferring the energy from the electron cloud around GFP over to the electron cloud around M cherry. So now we have two pathways out of here. And in this situation, we give off a red photon and M cherry falls back to its ground state. So the thing that you have to keep in mind is that we're always measuring averages and probabilities. Okay? So I told you earlier that that lifetime that we measure is the average lifetime. Each excited electron spends different amounts of time in that excited state. And what actually happens is the longer that molecule spends in this excited state, the greater the probability is that it's going to take this pathway out. Okay? This is the pathway of least resistance, so that's why it's more likely to go that way. But the longer it stays up here, there's a possibility of it heading over to M cherry. So that means now that our electrons that are staying up in this excited state for a longer amount of time are more likely to move over to M cherry, and we're not going to detect those on our detector because we're only looking in the GFP channel. Okay? So that's why our average lifetime that we're measuring for GFP is going to be shorter because we no longer have those long-lived lifetime molecules being detected by our detector. They're coming out as red photons. All right? So that's just what's written down here. So we're measuring averages and probabilities. The longer a fluorophore stays in the excited state, the more likely it is to fret, so to go over to the M cherry side. Therefore, the fluorophores with the longer lifetime return to the ground state via fret, and the fluorescence lifetime, which is just an average, is going to decrease, and that's why we see this shift in silhouette. Yeah?
So it means now you can measure the distance between the molecule, not just near or far? How sensitive it is?
Yeah, so there's, what you can measure is the fret efficiency. And actually with lifetime measurements, it's a very simple calculation to measure the fret efficiency. Perfect efficiency is 0.5, and that means these things are right side by side. So as your efficiency decreases, that means they're moving further and further apart. And there are equations that will allow you to do that math. You have to have your system that's very well characterized for both your donor and your acceptor, know basically the probabilities of passing that energy. And it has been done, but in the past when I've looked for this data, the numbers that you need for your donor and acceptor, there's very few of them out there that you can actually find. So I think for CFP-YFP, it needs to be published, but I haven't seen it from any others. I don't know how reliable it is. There's people that will certainly tell you that yes, it can be done, and they're much better mathematicians than I am, but it's tough to do. Okay, so earlier on, I mentioned that there are a number of problems with fret, and oftentimes I don't recommend it as a method to use in the facility. So I just want to take you through some of those problems that can occur and why Flim fret is a method to get around some of these. So here's our optimal scenario. We have two proteins of interest. They're tagged with two fluorophores, so one with CFP, one with YFP. And these two proteins of interest come together, interact with one another, and we get fret between these two molecules. So this is the perfect situation and what we're aiming for. Unfortunately, perfect doesn't always happen. So one of the things that can happen is these molecules, I mentioned earlier, they need to be within about 10 nanometers of one another, just about the size of a single protein. So depending on where you label your two proteins of interest with these fluorophores, when they come together, they might actually end up on opposite ends of that structure. And if that's the case, they're not going to be close enough to perform fret, and you're not going to get a signal, even though those proteins are perfectly interacting with one another. So in this situation, you get a false negative. Another thing that can happen is, if we go back to the previous slide, you might have them bound to your proteins of interest and oriented at the same side of this complex, but each of these fluorescent proteins has a dipole to it. So if we say the dipole is in the long direction here, these dipoles need to be lined up. So both molecules need to sit in this long direction side by side to actually get that energy transfer to happen. If these were still sitting side by side here, but one of these cylinders was lengthwise in this direction, you wouldn't get any fret because the dipoles would not be aligned. So these are two problems that can happen, and this is going to give you a false negative in any type of fret experiment. It doesn't matter which method you use. Now, something else that can happen is you can get an uneven expression of these. So here you can see we have a large expression of the protein tagged with YFP, but there's only very few CFP tagged proteins in the mix. And this, once again, with certain types of fret measurements, so sensitized emission or acceptor photobleaching, it's very difficult to measure this very small amount of fret that's happening in the sea of YFP. And there's a few other things that can, even if this molar ratio is closer to being even, things like fluctuations in laser power or changes in direct detector gain can also influence your fret measurement. The good news is these things do not apply to Flim fret, because you're measuring an intrinsic characteristic of the fluorophore, its actual lifetime, and that is not going to change no matter how many of these molecules you have there or what kind of imbalance you have. Another problem that you can get is homodimerization. So if your two proteins, either the actual fluorophores themselves, the CFP and the YFP, can homodimerize, or the proteins that you have them tagged to are able to homodimerize, this is also going to affect your fret experiment, and this will affect all types of fret experiments. And finally, if you throw in some endogenous protein that doesn't have your fluorescent tag on there, this can lower your efficiency of your fret experiments as well. Once again, this is not going to affect Flim fret, or not to the same degree that would affect sensitized emission or acceptor bleaching. And finally, another problem that you can have is the fluorescent proteins themselves cannot mature to a point where they can actually give off fluorescence. So you basically just have a lot of non-fluorescent proteins here, and when they pair up, you don't get fret. And this is going to affect all types of fret experiments. So I've told you the major issues with fret, and I've told you that Flim fret is able to circumvent some of these. The major one being that inaccuracies in the molecular ratio, or fluctuations in laser power, or changes in the gain on your detector. Basically the inability to make comparisons from slide to slide, or sample to sample, or day to day. But FlimFret does get around that, and the reason for that, as I mentioned earlier, is that intrinsic property of the fluorophore, its lifetime, and that's all you're looking at. So the biggest fluctuation to that lifetime is going to be temperature. So as long as you can get the temperature the same from experiment to experiment, your lifetime should be very consistent. But there is one other thing that can happen. When you're doing quantitative FlimFret measurements, so I'm not going to get into this today, but there is actually a way to say how many of my molecules with CFP are bound to a molecule of YFP, how many are free. You can actually make that measurement quantitatively. The really key to this is that you need a fluorophore with a mono-exponential decay. So that decay curve, when you're measuring your lifetime, has to be a mono-exponential. Not all fluorophores perform this way, but here is a list of a few of the fluorescent proteins that do. So that's, I'm just putting that up there for your reference. If you do want to start doing some of this quantitative FlimFret, how many free molecules are there versus how many bound molecules, where are the free molecules in the cell, where are the bound molecules in the cell, you can do these types of experiments quantitatively, but you have to make sure you're using one of these fluorophores with a mono-exponential decay. All right. And the last thing that I want to talk to you about with FRET is often sensors, intracellular sensors that are made by joining two fluorophores together work much better. The idea of having to bring two proteins together, interact, and get your CFP and your YFP close enough together to perform FRET is pretty difficult. But when you can make these into a single sensor in a single molecule, it becomes much easier. So here's a couple of examples of these. So there's a number of FRET sensors or FRET probes out there where it's just two fluorescent protein molecules that can FRET, joined by a polypeptide linker. The assay is there's some sort of protease that's expressed in the cell if something happens. This cleaves a site in between these two, and you no longer get FRET. Okay? Another example is two fluorophores joined with two binding domains. These are binding domains that recognize zinc. And when zinc is there, those two binding domains are brought closer together, as well as, and at the same time, they move the fluorophores away from each other. So this, again, you get FRET in the absence of zinc, and no FRET in the presence of zinc. And this can be done for a number of different ions. As well, here's another example of a binding domain for bile acids, and once that binds, again, you lose your FRET. And then one final example is a couple of epitopes for an antibody. Once that antibody binds, it gives some structure to this polypeptide backbone and moves those fretting proteins far enough apart that you no longer see your signal. So these are just a couple examples of intramolecular FRET probes, and these generally work very well, and there are a lot of them out there to sense everything from pH to CN levels, GM levels, things like this. So that's all I'm going to say about FRET. I'm just going to quickly show you a couple slides about some other applications of FlimFret just to finish up. So I mentioned to you earlier that you can do autofluorescence removal with FlimFret. So just like we do with spectral imaging on our confocals right now, where we take a spectra of the autofluorescence and we can filter that out from the spectra from our dyes. You can do the same thing if your autofluorescence has a different lifetime. So here, this is just an image of an arabidosis root that's expressing YFP, so the strong signal along the membrane here is YFP. But you can see there's also all of these intracellular little dots, background autofluorescence. And if we look at the image of the lifetimes, we see the YFP has a lifetime up around 2.5 nanoseconds, whereas all this autofluorescence has a very short lifetime. It's down in the blue region here, closer to 1 nanosecond. So what that means is afterwards you can basically just bin out or separate that short lifetime. And now we have two images here, one with our YFP that's been totally separated from our autofluorescence that's shown down here on the bottom. The other thing that you can do is actually look at autofluorescence and find different autofluorescent molecules within your sample. So on the left here, we just have a sample from the lime tree, and on the right, these are just standard culture cells, 3T3 cells. And this is just looking at autofluorescence after they've been hit with laser light. So I believe this is actually UV or near UV laser light on this side, and I believe this was infrared laser on this side. And what you can see is that you can start to differentiate different components of these tissues and these cells based on lifetimes. So you can see there's something in the nucleus of these cells that has a pretty long lifetime, whereas in the cytoplasm there's some components with shorter lifetimes. I mentioned TRPL before, so time resolved photoluminescence, same thing as Flim, but just the nomenclature looks more used in the materials world. So this is an example of cadmium beryllium thin film on the left here, and this is just an image basically looking for flaws or defects in that substance, and basically looking for a nice even autofluorescent lifetime image all over the sample, and where there are differences in this lifetime, you can infer that there are some defects in that sample. And over on the right-hand side here, this is just another image of some crystals, and essentially you can start to see different components of the crystal based on the lifetime. And finally, that autofluorescence, there are certain molecules within the cell that have been characterized to have a specific type of autofluorescence, a specific lifetime, depending on which wavelength of light you excite them with. So one of these is NAD, and here we have some cells that were either expressing high levels of the NAD kinase, or this was completely knocked out in these cells. And then there was a stimulation put on the cells to stimulate the production of phosphorylated NAD. And so what you can see is that this NAD molecule, when it's in its resting state, has a pretty long lifetime, so up around 4 nanoseconds. But once that kinase phosphorylates it, the NAD pH, it now has a much shorter lifetime, down in the 2 to 2.5 nanosecond range. And this is actually a method used to image intracellular metabolism in a label-free method. So there's actually no label in here, you're just looking at the lifetime of the autofluorescence of the NAD. So finally, just to remind you, we have 2 microscopes in the facility that will be capable of doing this. So on July 15th, the Flim equipment is going to be installed on 880 number 2. And here we've got pulse laser lines at 405 and 440 nanometers. And this fall, probably in mid to late October, our new 2-photon system is going to be arriving. This will also have a 440 pulse laser on it. And then we will have a tunable infrared laser. And this is actually incorrect. This should actually say 700 to 1300 nanometers. It's going to be the new insight laser from spectrophysics. And so this should be arriving, like I said, in the fall, in late October. Great. So once again, if you have any questions, feel free to send me an email or drop by my office. You can also head to our website where we'll be posting all of these seminars from the summer. Thank you very much.